This is the last recording for Chapter 2. I try not to make the recording so long, but it's just a lot of information in this chapter. So we're going to talk about proteins. We've covered carbohydrates and lipids. There are 4 groups of organic molecules that are important in the human body, and they are carbohydrates, lipids, proteins, and nucleic acids. So we discussed carbohydrates and lipids, and if you remember, they both contained the elements carbon, hydrogen, and oxygen. Proteins contain carbon, hydrogen, oxygen, nitrogen, and then some sulfur and phosphorus. Mostly sulfur. DNA contains more phosphorus and proteins contain more sulfur, but both contain nitrogen. The building blocks of proteins are amino acids. So we say amino acids are the building blocks or the monomers of proteins. There are 20 different types of amino acids. I was going to see if there's a picture of an amino acid, but I guess not. No. Okay, so an amino acid is going to contain basically a central carbon, and that carbon is bonded to an amine group, a carboxyl group, and an R group. And the R group is not, R doesn't stand for an element, it just is a variable group. So all the 20 different amino acids, what makes them different is their R group. Okay, I really can't believe there's not a... Let me try this because I think you need to see the chemical structure for proteins. Okay, so maybe I can do this. I'll do my best. So basically, a nucleic acid has a carbon, a central carbon, and that carbon has 4 groups attached to it, like that. So one group is just hydrogen. It's just a hydrogen atom. Another group is the R group. And then I don't know that I'm going to be able to draw these, but the carboxyl group is a C. No, I'm not. I don't do a good job with. It's a C double bonded to an O, single bonded to an OH. It's called a carboxyl group. So this COOH, just try to ignore that little piece that shot out there from that C. I don't know why that happens, but every time I try to draw something, it does that. But anyway, there's a central carbon bonded to a carboxyl group, that's the COOH, and then also bonded to an amino group, and that's nitrogen and 2 hydrogen atoms is your amino group. So this is what makes it an amino acid, is that it has an amino group, and then the carboxyl group is an acid. It makes it an acid. So you've got the amino group, the R group, the carboxyl group, the hydrogen group, and that's what an individual amino acid looks like. Okay, well, that wasn't so bad. Then, as far as the functions of proteins, it's easiest just to say that proteins have a lot of different functions. Some proteins are hormones, some are enzymes, and so those hormones and enzymes function in regulating processes. Proteins can transport, for example, the protein hemoglobin in our red blood cells transports oxygen. Proteins can provide protection They can help muscles contract, like the proteins actin and myosin cause muscle contraction. Proteins can provide structure. They are part of the plasma membrane, and they can provide energy. So we have the primary proteins have 4 levels of structure. The primary structure of proteins is just the chain of amino acids. So a protein can have 1,000 amino acids, or it can have hundreds of thousands of amino acids. But those amino acids are bonded by a chemical bond called a peptide bond. It's a special type of covalent bond where the amino group of one amino acid is bonded to the carboxyl group of another. But the primary structure is just the chain of amino acids like you see here. Just chain of amino acids that forms the primary structure. Then the secondary structure is either one of two things. It's either a pleated sheet or it is a helix, like a spiral shape. So that chain can either form into a pleated sheet or it can form a spiral helix. And then the tertiary structure is when the pleated sheets and the spiral helix chains fold even more into a 3-dimensional shape. Okay, so the secondary structure forms from hydrogen bonding within the chain of amino acids, and the hydrogen bonding creates either pleated sheets or beta pleated sheets or alpha helices. And the tertiary structure comes from folding that occurs by interactions with the polypeptide chain and its environment. So basically it's just folding into a 3-dimensional shape. And then some proteins have a quaternary structure, and that means that there is more than one tertiary structure bonded together. So insulin, for example, is made of 2 polypeptide chains, so it would have a quaternary structure. Those 2 chains would be bonded together. Hemoglobin has 4 polypeptide chains. So again, the primary structure of a protein is just the sequence of amino acids in the chain. The secondary structure is either a pleat or a helix. And the tertiary is the final 3-dimensional shape. And if it has a quaternary structure, each color represents a different tertiary structure. So it's just more than one tertiary structure or more than one polypeptide that is chemically bonded together, and that would give it a quaternary structure. Other things that are important about proteins is proteins can denature. In other words, their shape will change, and they aren't functional when their shape changes, because the structure of a protein is very important in whether it will function properly or not. It has to be exactly the right structure, and when that structure is broken and when that structure is changed, then the protein is denatured and it doesn't work anymore. So some factors that can cause proteins to denature are extremely high temperatures and also the wrong pH for that protein. Enzymes are protein catalysts, and what they do is they lower the activation energy for chemical reaction. The way they work, if this yellow molecule is your enzyme, is that the substrate, molecule A, will bind to it, and then the enzyme will, well, actually, there's 2 substrates. I apologize. Molecule A and molecule B are the substrates or reactants, and they will bind to special regions on the enzyme called active sites. And then the enzyme and the molecule A and B will together kind of change shape a little bit. And then, as you can see in this case, the enzyme catalyzes the chemical bonding of molecule A and B. So now we have one molecule formed from molecules A and B. We just call it AB. And it's released from the enzyme, and now this same enzyme can catalyze more reactions between molecules A and B. Now, our last set is going to be, we're going to talk about the nucleic acids, which are DNA and RNA. There are two types of nucleic acids. There's deoxyribonucleic acid, and there is ribonucleic acid. All nucleic acids are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the building blocks of nucleic acids are nucleotides. So nucleotides are the building blocks or monomers of nucleic acids, and they are composed of a nitrogen base, a phosphate group, and a 5-carbon sugar, either ribose or deoxyribose. The nitrogenous bases are either single ring molecules, thymine or T, cytosine or C, uracil or U, or they are double ring molecules. Adenine is A, guanine is G. The DNA structure is a twisted ladder, which is referred to as a double helix. The sides of the ladder form from the deoxyribose sugar and phosphates. The sugars and phosphates form the sides of the ladder, and then the steps or the rungs of the ladder are formed by the bases. So in every step or rung, if one of the bases is an A, the other one's always a T. A only pairs up with T. And if one of the bases is C, then the other one is G. C only pairs up with G. So DNA has 2 strands, and that is why we have the pairing of bases. The steps of the DNA ladder are formed by 2 bases that have paired with one another. A only pairs with T, C only pairs with G. But RNA is a single strand molecule. And the only difference with RNA is that instead of having a T, the RNA will have U in place of T. And this is what the structure of DNA looks like, a twisted ladder or double helix. And you can see phosphate, sugar, phosphate, sugar. The sides of the ladder are just alternating phosphates and sugars. And the steps are the bases. So you can see the base T is paired with A, G is paired with C, A is paired with T. The A's and the G's have 2 rings, and then the T's and the C's have 1 ring. So that means the steps are a total of 3 rings wide. So that's basically the structure of DNA. One individual nucleotide would contain a base, a sugar, and a phosphate. Like that's a nucleotide. So we have 1, 2, 3, 4 nucleotides shown in the strand on the left, and then we have the nucleotide on the right is turned upside down, but it's still the adenine and the sugar and the phosphate. That makes up a nucleotide. So we've got 1, 2, 3, 4 nucleotides shown on the right. So there's a total of 8 nucleotides in this picture. RNA is not a double strand, it's a single strand. So when we're comparing RNA and DNA, RNA is single-stranded. RNA kind of helps DNA in making the proteins that contribute to our traits. So RNA helps to express the genes that are found in the DNA molecule. But RNA is a single strand. The sugar is ribose instead of deoxyribose. And the bases are A, U, C, and G instead of A, T, C, and G. And the last molecule we're going to talk about is ATP, adenosine triphosphate. And this is the molecule that all living organisms use for energy. It is often called the energy currency of cells. Because our cells produce ATP and use ATP as a direct energy source. ATP has 3 phosphates that are bonded, and when ATP breaks down into ADP, adenosine diphosphate, plus phosphate, that energy is released and is available to do work. ATP looks a lot like a nitrogenous base because it is. ATP is a modified nucleotide. It's an adenine, a ribose sugar, and 3 phosphate groups instead of just one. So that's why we include it in the nucleic acid section.